welcome to the Preemie Chats. Thank you all of you for joining us here today. I'm Fabiana Bacchini, Executive Director with the Canadian Premature Babies Foundation. CPBF is a charitable organization, and our mission is to support and educate families of premature babies. This Preemie Chat series is one of the many initiatives we have to bring information for NICU families and healthcare professionals. Here every Friday, we talk with experts, researchers, and parents who share with us their experience and knowledge. Also on our website, canadianpreemies.org, parents will find all kinds of resources and support. And today we're going to talk about what is new in the maternal milk. Human milk is precious for the growth of preterm babies. In particular, helps the maturation of their immature gastrointestinal tract and has a strong immunoprotective effect. Researchers still don't know all the golden properties of human breast milk. With this presentation, we'll bring parents up to speed on new discoveries and learn what tiny, powerful breast milk molecules do to positively impact long-term health outcomes. And I have here with me joining us live from Ottawa, Dr. Emanuela Ferretti, who is a full professor of pediatrics at the University of Ottawa and a neonatologist at the Children's Hospital of Eastern Ontario, CHEO, and the Ottawa General Hospital. Since she joined the University of Ottawa in 2007, she has been a co-leader in implementing an innovative neonatal ethics teaching program focused on parent-physician communication skills for postgraduate trainees. Her research interests include an ongoing multi-center CIHR-funded study on the development of the immature human intestine and the genetics underlying necrotizing enterocolitis, as known as NEC, as well as the impact of different molecules and drugs on the premature gut. Dr. Ferretti, thank you so much for joining us here today. I have to tell you, I'm very excited to learn more from you and more from your research.
Thank you so much, Fabiana. First of all, I really thank you so much to have me. Also, I really thank you, Fabiana, because I'm so grateful that for your relentless advocacy, really, in connecting people that are working in different, spheres and in different domains for the well-being of our babies today and also for the future of the well-being of premature babies and their families. Thank you.
Thank you. So we are going to have your presentation here up on the screen. Give me just a moment. And for all of you watching us live from YouTube or Facebook, please send us your questions, your comments. We'd love to hear your stories, and we're going to address them at the end of her presentation. So the floor is yours.
Okay, thank you so much. First of all, I would like to talk to you about what's new in maternal milk. Probably you know, but I want also for you not to be intimidated about what you're going to see. It's very complex. It's not easy task, but it's so exciting that I could not share with you. So next slide. Who can't see that? Sorry. Yes, so I'm going to do the outline. No, it's okay, Fabiana. Yeah. So I'm going to do the outline quickly. We're going to have a quick overview of the spatial tiny molecules called, exosomes that are present in the human maternal milk. Then I'm going to talk to you about my research finding, about my group here in Ottawa and in Sherbrooke and the impact. And then we're going to have a quick peek into the future.
Okay, give me 1 second because the slides are not moving. Just 1 second, please.
Sure.
Okay, let's try this again.
Okay. So we were at the outline, yeah. And then you can go to the next slide. Okay, so human breast milk is essential right after birth and is an irreplaceable source of nutrition for early human survival. Now, various infant formulas have been developed for greater similarity to human breast milk in terms of many components like, carbohydrates, fat, and proteins. And overall, we acknowledge that there is no perfect substitute to human milk. Why? Next, because we are learning that in addition to nutritional biomolecules, the human milk contains immunoglobulins, microbiomes, next, extracellular vesicles, like exosomes. And it's about this exosome that I would like to talk to you today, because there is evidence that is directing to the fact that non-nutritional bioactive components like this one, they impact profoundly the health and the survival of infants. For example, next, breast milk was identified as a very rich source of microRNA. And microRNA, imagine like they're little bricks that they build everything inside the cells. And they really play a very important role in the survival of many biological processes. And other associated benefits of breastfeeding include, next, include the growth and the development of, the babies, the protection against pathogens, and therefore immunity development of the baby, the promotion of gut colonization, and all this allows to prevent gastrointestinal diseases. Next. So if we look at the research of the exosome, these little molecules, it's really funny because interestingly, the exosomes were originally regarded as cellular metabolic waste. So garbage that the cells were really dumping into our system. But then they got their own name in 1987. And then somebody discovered that in 1996 that actually the B lymphocytes were responding with exosomes to anti-tumor factors in 1996. And also in 2007, other researchers found that actually there was an exchange of genetic material between cells through these exosomes. So you can understand that at that point, we were about, 2007, people realized, they became aware that actually the exosomes were very, very important and became, since then, hotspot research since 2007. Now, if you look at the story there, if you look there in 2013, you can see that, some researcher also got a Nobel Prize because they were able to prove that these molecules, actually, they can be used to create anti-cancer vaccine in the future. Next. So in 2016, with my group in Ottawa and in Sherbrooke, we were I was working for a long time, for many, many years, on biomarkers of necrotizing enterocolitis, which is a very ominous intestinal disease that attacks particularly preemie babies. And so we decided that if we got funded by the CIHR, and we started to look at this molecule inside the breast milk, the human breast milk. Next. So what are these human milk exosomes? There are mainly parcels that protect from degradation lots of goodies, in particular proteins, lipids, messenger RNA, microRNA, and also DNA. And next, these vesicles are also, if you look at the microRNA inside there, they're involved all in, in development, in differentiation, in creating new cells, in adjusting the metabolism, and also they do, in a sense, lead to death of cells and tissues. Next. So when we look at about the size of these molecules, the fun part is that actually the exosomes, as you can see, it's a teeny, super tiny molecule that actually has the size of a virus. So the teeniest molecule that we can, with what we have now, that we can see in a biological system. Next. So, and how this exosome function? So these parcels that we have discussed, they function as a cellular communication. They do imply a method of listening in to the crosstalk between a mother and an infant. Next. And breast milk has changed from a simple food to a highly sophisticated maternal neonatal nutrient and non-nutrient communication system, orchestrating early programming of the infant. Means that what we're going to do and discuss today between me and my mom that is feeding me is going to make me the future person that I'm going to be later on. Next. Indeed, a fun fact in 2016 is that there is a component of regulation that research has studied and then figured out that these microRNA are diet specific. Means that each mom, they put lots of mom together and they check their milk while they were breastfeeding their son and daughters. And what they discovered is that there were specific, for each couple of mother and baby, there were specific microRNA that they were targeting the needs of the baby. And this is super fascinating, isn't it? Next. So the other function that the exosomes do as biomarker, they are, they are involved in connecting and talking to cells. So as you can see on the left, there is a secreting cells, and next, there is a teeny tiny part of the exosomes that you can see now. And this exosome is going to be taken up by the recipient cells. And this means that let's say the secreting cells secreting cells from the duct, from the gland, the mammary gland that produces milk, and the recipient cell on the right, it could be a cell from the gut, from your baby, from the baby that you are breastfeeding, and this is very important because biologically. The health of the babies is going to be very much impacted by it. Next, when we look at the publication history for extracellular vesicle and exosome, we did a review because we have worked for many years about the characterization and isolation of these molecules, micromolecules. EV stands for extracellular vesicles because it's part of the family of the extracellular vesicles. Now, when we look at the publications, what we found out is that some people from the 1950s started to work on milk extracellular vesicles. But if you look at when they started to work on the human milk extracellular vesicles, we go only from 2007. And then again, only a quarter of the publications are, the one that you see in orange, is related to studies and research on human breast milk. Same thing in figure B here. You're going to see that this is the microspectroscopy and studying on the MS, also in pink. There are not very many. And again, it's a very small part of the scientific research right now. Still, next. So when we started in 2016, my research group was committed to investigate and understanding the mechanism by which human milk benefits the infant. And this is what we knew at that point, that these microRNA were abundant in all body fluids, in the blood, in saliva, in urine, and in amniotic fluid. But the most abundant content of RNA and microRNA has been in the human breast milk. There are about 1,400 different species of this microRNA, and all of them, each one of them, is targeted to a specific function in the cell. Also, we found that there was no consistency in the literature on methods for exosome isolation and RNA extraction from human milk. And so in the next slides, you're going to be, and as I said, don't get intimidated because I'm going to guide you. But, I wanted to highlight some of the research findings that our group were able to see. Next. So our first objective, as you can see, was to collect the milk, fresh and stored, and then isolate the exosomes from it. Next. And when we tracked down the human milk exosome, yes, we found that these nanoparticles are found both in term, as you see the blue line, and also in preterm milk. And also we were, within the expected size. When we look at the scan electron micrograph, we found white pearls. Well, this is how I call it. I found white pearls, which is really an exosome that has been isolated from a small sample of human breast milk. Precious. Next. When I then look at them suspended in a solution, I really found fascinating that I found a universe of planet. And so when I look into the microscope, you can see on the left side, this is how the exosome look like when I use fresh human milk. And they are shining and they're blinking. They're really like stars and planets. Then on the right side, this is how I look them, how I see them, when I isolated after freezing the milk for 24 hours at -20. So in our regular freezer, right? Next. Then in this, next, please. Thank you. So then in this starry sky, when it's fresh, I see that there are single particles and multiple overlapping particles. And then when I look at the exosome size, again, I see expected size of molecules. Next, when I look at the starry sky on frozen, I see that there are different size of multiple size of particles. Again, I see larger exosome size within the same range, and so this reassures me that both, in frozen and fresh milk, I can recognize these particles. So next slide, please. So in conclusion, the conclusion of the first part of the experiments that we did is that the concentration of these microparticles from the frozen milk sample was a bit lower than the one of the fresh milk. Maybe due to the fact that while we are freezing, we are breaking down the lipid membrane. And then we demonstrate a difference in abundance of exosomes depending on storage procedure. And so we concluded that the storage of breast milk can alter the exosome population. Next, please. Now, going back to this, how does a human exosome look like? Because, there are animal exosomes, but, we were looking at human exosomes. When I first saw how an exosome looked like, I was completely and totally fascinated by it. And actually, I thought, the first thought I had was, which planet is this one? And, just to summarize it for you, so if you look at the crust, imagine that this is Earth, right? You look at the crust and it's a double lipid membrane, which is protective to what there is inside. And inside what we can see, there is a mRNA and a microRNA. Then, if you look at the surface, I thought that this one looking like skyscrapers. But what we call them, these skyscrapers, we call them protein surface markers. And why they are important? Because each exosome has different skyscrapers, right? And actually they can be distinguished from an exosome that comes from, for example, a cell versus another, like a cell from the liver. It's different. It's going to give me different skyscrapers than an exosome that comes out of a cell from the kidney, for example. Next. Yeah. So we are going to talk in particular about these skyscrapers that are called theta-spanning. Next. And what do the theta-spanning? I call it mending proteins because they look like mending, through the membrane. So as you can see, these are, they take their name because they have 4 domain, we call it, that they cross the membrane. See the gray part here on the right side of the picture. This is the double membrane of the exosomes, and they go in and out like a mending, right? And they are proteins that they really regulate the cell mobility and motility. They can even regulate the cell shape. They can regulate the signaling between, remember when this exosome goes from one place to another. And also they change the plasma membrane dynamics. And mainly they are regulating cell protein trafficking. So these skyscrapers, they close the door and they say to the cell, no, you cut dear nucleic acid, you stay here because I don't need you to go out. Stay in there until I find the right place for you to go. And so it's really fascinating to see how these molecules, they have these characteristics, right? So obviously, taken by curiosity, next slide, please. We look into, by using flow cytometry, we look at which kind of skyscrapers, so which kind of marker proteins were isolated in the term and preterm maternal milk exosomes. And what we found, as you can see in the blue line here, in the blue circle, we found all this list of skyscrapers that were common between term milk and the preterm one, which is delineated by the red line. But also, surprisingly, we found that there were unique proteins belonging to preterm milk exosome only. That you see them, the only one that are there in the red balloon. Yeah. So next slide. So when we look at the mainly when we wanted to see at when we wanted to trace this and capture this, we call it CD9 because this is what the type of skyscraper that we were looking for that is very, very important. When we wanted to look and capture, we saw fireworks literally, and in particularly, if you look at the preterm picture, the left picture, you saw a lot and very much abundance of this type of proteins produced by the exosome compared to the one that you can see clearly is less shiny in the full term. So the fact that the preterm milk exosomes has abundance of this skyscrapers, these receptors, right, whatever, these proteins indicates that really the maternal milk try to enrich the exosomes of this little protein because they hope maybe that they're going to be caught, they're going to be taken by the intestinal cells, the cells in the gut of the baby. Next, please. Now, as you can see, when we did the characterization of the total protein cargo on the cargo of, inside these parcels, we found that there was a huge difference between the milk of mothers who gave birth to term baby compared to preterm babies. And if you look at the picture, you see that the milk exosomes contain much more proteins than when they were preterm, when the milk was a preterm milk versus the term milk. And this suggested to us that the production of the milk response is really in response to the shock of the infant that is born too early and creates a so-called magic milk, since we don't know exactly, unfortunately nowadays still, what these components do. But it's a very interesting question, right? What exactly this protein or those proteins do? Next slide, please. And so we had a question, right? So how does the composition of in preterm milk exosome differs from the term to help the preterm infant to survive, to catch up their growth and thrive? Next, please. So when we investigate at the differential composition of these proteins, right, we found different profiles, protein profile. Next. So first of all, we found that we were able to characterize 155 proteins, unique proteins in term milk versus 287 unique proteins in the preterm milk. Next. And while in the term milk, these unique proteins are categorized just to metabolic processes, this made completely sense for us because metabolic processes are important for growth of infants, which is the primary goal for a term healthy baby to be, in terms of needs. Next. But when we try to highlight what the premature milk, exosome profile was going towards, we found that actually these exosomes, this protein, were biologically involved with the development of the structure of the anatomy. Means they are involved in all this biological process that they make the baby grow, make the cell grow, make the liver grow, make the muscle grow, the bone grow, and then they regulate the gene expression. Also, we found that they respond to other organisms, like, for example, they do respond to bacteria or viruses, right? They have the capability to respond for axonogenesis, which means the development of the brain cells, and also regulation, as you can see in the little ball on the right, of hemopoiesis, means all the blood cells, especially the red blood cells and the platelets that are contained in our blood. And this made totally sense. Next, because the fun fact is that when we looked at the exosomes that were present, the protein, right, of the exosome that were present in the amniotic fluid, and this was work that has been done by other researchers in the world, we found that those were the same exosomes protein that they were prevalent in utero, which makes sense because a baby that is born premature is born too soon, is not supposed to be out, is supposed to stay in utero. So we were very happy to find these findings. Next, please. So the other conclusion that we figure out is that we identify unique exosome surface marker proteins of preterm maternal milk. Now, the term milk exosome unique proteins are involved in metabolic process related to growth. The preterm milk exosomes are involved in development and gene expression regulation. So we can, in a sense, speculate that the human milk exosomes are equipped with unique proteins that could aid the catch-up developmental process for preterm infants. Next slide. So we went on At this point, we were totally in it, intrigued. I mean, we had tons of questions, but we needed to go slowly and step by step. So the next step we wanted to do, when we had characterized the exosomes and purified and isolated properly, it took us about 3 or 4 years of work. We wanted to characterize the microRNA, and with this from both term and preterm, and also fresh and store, because we wanted to see what happened to the RNA. Next slide, please. And when you look at you have a blue and a reddish. So the red is the fresh milk exosome RNA, and the blue is the RNA sample from the frozen milk exosomes. Now, when we did the quantification, as you see here in the on the right side and the right upper panel, we found that there were larger nucleotides, means these little bricks that they compose, the mRNA, the microRNA. They were abundant and there were a lot. When we did look at below in the blue slide, in the frozen milk, we found that yes, they were there, but they were many more particular, like we saw in the starry sky before. I don't know if you remember. And probably this was due to the fact that we had an accumulation of dead cells, because when we freeze, cells die. And so probably that was due to that was the reason why we found a little bit more RNA in that. But still, if you look at the quantity of the overall exosomes, we were able to find them. Next slide, please. At this point, what we did, we had fun and we looked at the big red bar. So it was fresh milk with the fat. Then we did it on fresh milk, skim milk without the fat, and then the exosomes. And then you can see there is a decrease in this, in the RNA content as when you manipulate the milk, they're still present. The RNA is still, but obviously you lose it in the process. Same thing again for the RNA of frozen, like the blue line. Now, if you look at the frozen milk fat from fresh to frozen, you see that it was about half. And probably this is because in the fat, there is a little enzyme that dissolves the RNA. Still, we were able, if you look at the two things on the right, at the two graphs on the right, we were able to characterize exosomes. So the exosome was still present. And so next slide. At that point, what we wanted to do, we did more analysis and we found 9 abundant microRNA in fresh milk human exosomes. And then when you look at this, it looks like almost like a firework, is a network, is a targeted network that tells us where the microRNA are implicated, and they work with different, diseases, inflammation, creation of new cells or destruction of new cells, etc., etc. And what we found is that all these 9 abundant microRNA in fresh human milk exosomes are connected to mainly to inflammation. So you can go to the next slide. So the storage of the milk, we concluded that the storage of the milk can affect the RNA population. The concentration of the RNA isolated from these little microparticles and the length is also altered by freezing milk for 24 hours. And obviously the crosstalk between a mother and an infant provides for the protection against inflammation. So we wanted to believe, and we strategized also, looking at the other people that were working in the world, that our conclusion were the same as other groups were, few groups were doing. Next, please. Now, by now, when you look at this, you should be knowledgeable, knowing that in the breast milk, there are these exosomes, which is, I make it big for you, that they contain a lot of goodies and they go into the intestine of the babies. Now, obviously, if a baby is, one parenthesis is that, remember that when I was, in my group has worked for many years on necrotizing enterocolitis, NEC for short, which is the more severe disease of intestinal immaturity. And so obviously we were interested to see the implication, right? So what we know is that when an infant is born at term, the cells and the structure of their gastrointestinal tube are ready, are mature, and there is no problem. Now, we know that unfortunately, the population that we have now is not the same as we had before. Now, there is a worldwide increase in prematurity. And obviously, when we have a premature gut, it's not the same, right? Is fragile. We give them antibiotic, and so we destroy the good, the good microbiome, the good pathogens that they have there, the good, bacilli that they have in their gut, and they make them more susceptible to NEC. So if we look solely at the nutrition cause of NEC, we know that there were many studies conducted on cohorts of newborns in which they show that the incidence of NEC is increased about 10 times if you use formula milk. And instead, a meta-analysis that looked at 4 clinical trials concluded that infants who received human donor milk were 3 times less likely, as you can see in the picture here on the left, that there is a decrease of 3 times the likelihood to have NEC if the babies were fed with exclusively human breast milk. Next slide, please. So again, don't get intimidated. It's a fun slide. It looks very complicated, but it's not. Just follow me. So unfortunately, I don't have the pointer, but the first thing that I want to tell you, and the first fun fact that we know, next slide, please, is that the exosomes, when they are swallowed by the baby, they go into the stomach, and then they reach the intestine. It's interesting because this exosome, they resist the harsh intestinal environment. So even if you have acidity in your stomach or whatever, they don't get destroyed at all. So they reach the mucous barrier, and they reach the intestinal cell, and then they go through. So the idea is that the microsome, see, you saw it here, milk exosomes, can you see it? The round double layer thing, goes into the epithelial cells, the cells of the intestine, and instead of going through directly, first interacts with all the other cells. So they interact with the goblet cells, and the goblet cells, they increase the mucus, and so the barrier against bad bacteria. Also, they interact with the intestinal stem cells that you can see there. And so what they do, they increase the regeneration if there is any breakage or anything that goes wrong. They help to the intestine to regenerate. Also, what they do, they ask the cells to proliferate, just to multiply. And then they have also an anti-inflammatory activity, and also they tighten up the barrier by acting on the outside of the surface of the cells. And then in between the cells, when you see these yellow dots, So these are called tight junction, and they close it. They help to close it. So they do a lot of stuff before going down there into the intestinal capillary and reach the systemic circulation of the baby. Now, we still don't know a lot about what, once they are in the system, the circulation of the baby, where they go and what they do. But there is at least one study that tells us that once the exosome is in the blood, they reach the cells in the brain of cells that is in a center that regulates the food intake and the adipogenesis, means the storage of fat, which makes sense, right? Because this is the autoregulation of breastfeeding your baby, and the baby knows when he's hungry and when he's not. Okay, next slide. Quickly, I just wanted to tell you that in 2010, this is the first group that look at the milk and look at the milk in terms of, they found that the breast milk in the first 6 months of lactation can modulate the development of the infant's immune system. Next slide. And 2 years later, we had another group that from China that they proved that there was a transferable genetic material from the mother to the infant. So next slide. We did, the question here looks very complicated, but mainly we ask, okay, when the exosome reach the intestinal cells, what do? Do they change the genetic of the cells? And so we went ahead and we did another experiments. Next slide, please. So what we did, we took again the exosome, isolated, we put it on a, you see here, there is a layer of CACO2 monolayer cells, which are a special, a human intestinal cells. They resemble very much so the intestinal cells of a baby. And we put exosomes at 0, 6, 12, 24 hours first, and then we're going to see the results. And then the second part of the results, we put them on some bad bacteria that we usually found in the gut of baby that they have neck. And we wanted to see what was the response to the inflammation. Next slide. So the very first thing that we saw is that when we incubated the cells at 24 hours, look at the 24 hours panel here, this light and blue, when we pre-treated the CACO cells with the exosomes, they increased a lot compared to 6 hours or to 0 hours, which makes us think that, yeah, the exosomes that we have isolated, indeed, they ask the cells, the intestinal cells, to multiply, which is a good thing. Then, next slide. So next slide is telling us that what we found is that also, but these are preliminary results, so still, to come better results. But when the CACO cells were pre-treated with term or preterm milk, they decreased. Look at the number 1, the A slide, the A figure. So here we were looking at the marker, which is IL-8, is a marker of inflammation. And usually if you are very inflamed, it goes up. If you have no inflammation, it goes down. And what we found is that when we did pre-treat with exosomes the cells, we were able, as you can see, compared to the white bar that is the control, we were able to decrease the level of this pro-inflammatory cytokine. Then what we did is something that made us more confused, but again, promising. When we put the whole milk, you see that in the figure B, there is the whole milk and the control again. When we put the whole milk, so not only the exosome, but just the fresh milk, and we put it on the cells, we had an increase of this cytokine. There are lots of theories. We don't have the time to go. Maybe you're going to ask, but it's interesting to see that we still have further investigation that needs to be done. So next slide, please. In conclusion, so in vitro treatment of intestinal cells with term or preterm human milk the exosome attenuate the inflammation. We can say that now. Also the cargo, of the human milk exosomes, they have a potential therapeutic target for infant diseases, especially I'm thinking about the gut. And the Mirna in fresh human milk exosome relate to inflammation, and they demonstrate also that the mother provides further protection because increases a lot of microRNA that they can protect cells from it. Next, so few practice points. So human breast milk is the only reliable source of biological active microRNA. In the current artificial infant formula, there is no detectable physiological levels of microRNA or milk exosomes, so we couldn't find them. In the bioactive exosomal Mirna of milk, are this micromolecule, these exosomes, they are eliminated either by boiling or if they are in front of microbial fermentation. But pasteurization does not kill this microRNA, so it's very important. Next slide, please. So the storage of breast milk can alter the exosome population, and in turn can also, kind of alter the microRNA message that are being delivered to the intestine of the newborn. But one good thing is that we figured out that there are unique proteins in human milk exosomes that are really, really unique for a unique developmental needs of the baby in these maternal infant diets, which is amazing. And then human breast milk is so highly complex system of various bioactive components that is indispensable in the formation, we do believe is indispensable in the formation of early immunity. Next, please. So for future, we are going to monitor more inflammatory response using pretreatment with preterm milk exosome, and we're planning to do other study with other human cells. And also keep in mind that for the future, we are thinking about a possibility to use this exosome to develop either a drug carrier or a microRNA carrier, if it's particularly beneficial, directly into the intestinal cells. So as already been done there is a lot of pharmaceutical companies that are looking for target anti-cancer, therapeutics. And next slide. So we do believe that this research seeks to tackle the challenging search for new prevention and better diagnostic and therapeutic strategies for neonatal intestinal diseases like the necrotizing enterocolitis. When I showed this slide to my PhD, she laughed and she said, Dr. Ferretti, every time I do an experiment, I feel like that. So the challenges are ahead for sure. Next slide, please. I would like to thank you, everybody. First and above all, and sorry, but because Fabiana doesn't have the last slides that I sent her that I prepared, but I wanted to thank you, particularly the mothers that they so generously donated, despite their stress in having a premature baby, the milk to us, to our group, to do the studies. And I wanted to thank, obviously, my double collaboration here in Ottawa with Dr. Althusser and Brett and Jamie that are the PhD that are working on it. And on the other side of the University of Sherbrooke, Jean-François Beaulieu and Eric Tremblay. Also, I would like to thank our research assistant, Becky and Chantal, because they are going to the mothers and they ask them if they would donate some milk for us. And I think I'm ready for your questions. Thank you to all.
Dr. Ferretti, thank you so much for your presentation. It was fascinating, very intriguing, and I have some questions, but also I want to open with a comment from Shireen. She said, marvelous results. In Islam, those who breastfeed from the same woman become brothers and sisters and forbidden to marry each other. Now, exosomes explain why. Really interesting. Thank you, Shireen, for this comment. I really appreciate that because it really brings us all together to be more understanding and sensitive to different cultural backgrounds. And I'm always fascinated to learn and see how can we move on together. My question to you is, having this knowledge that you have now, how is this going to change practice?
That's the $1 million questions. Well, first of all, I would say for sure from the very beginning, because we know, and I think your session on breast milk and when you did the breast milk week and everything, for sure it's very important to talk to mothers and let them know that their milk is so precious. It's very, very precious. And so even if it's in a situation in which they have very little, doesn't matter. We put it in the mouth of the baby. The immunoglobulins, they go from the mouth directly into the stomach and they coat the intestine with this immunoglobulin, which is very important. So very proactively, try to strategize and try to have fresh breast milk as much as we can for both term and preterm, especially preterm babies, obviously. And then the second strategy is that there is donor milk also that can be used, definitely. So pasteurization, as I told you, does not kill these exosomes. And so in the donor milk, it should be, it is not been boiled before, but they should be this precious carrier and with microRNA that, they really can induce a lot of biological activity, precious, and really start to form the immunocompetency, we call it, of the babies that they are premature. And then obviously advocate, like I'm doing for many, many years now in Ottawa, but advocate with parents and et cetera, about formula that are exclusively human milk based, not bovine milk based.
Okay, so you touch on different points and I have different questions from some of those. So the first one is fresh versus frozen.
Yes.
You saw the benefits on the fresh human milk to the baby. And as far as I know, many NICUs still freeze the milk before giving to the baby. So is this a way for you to see that it's going to change practice in the future, seeing the benefits of fresh human milk?
That's a very good question. I still want to do some experiments and try to go ahead and see for how long we can see exosomes when we freeze a sample. Because the point is that what we know is that we can freeze it up to a certain point and then there is a degradation of the proteins. But here we are talking about something a little bit different. We're talking about microRNA. It might be possible that even, in freezing, for a certain extent of time, these exosomes are still present. These precious microcargo, they are still there. We don't know yet. Nobody has done it yet. Maybe I'm going to do it. That would be good. Obviously, the advantage of the fresh milk is that, as I show you, there is a little bit more microRNA. And there is a little bit less dead cells, and there are lots of exosomes with a good length. They have a really good length of all these little bricks. They're a little bit longer than the others, but the point is we want human milk.
Right. So my next question is, because you talk a lot about the unique, the specific for the mom and her particular baby.
Yes.
So from what I understood a little bit of your work, is this a possibility in the future to have a match donor that was going to match that mom's human milk? Like another researcher mentioned that once, and I was totally off my head. But now when you talk about this, is this a possibility? This is where we're going with this kind of research in the future.
So that's a very good question. You should be part of my research team, by the way, Fabiana. But, I think it's a very intriguing question. So has already been proven by other, the study that they found that there was a targeting mRNA and DNA, that actually the mother with the baby, they interact with each other. And then in this group of mothers, so they look at the milk before being extracted and after the breastfeeding, okay? And they follow the mother for 6 months. And they were able to see that the mother of that specific baby was giving special and unique mRNA to that baby, because that baby maybe needed to, I don't know, maybe he needed to grow the kidney faster than the intestine, or the brain instead of the gut. Who knows? We are still at the beginning of this. Really, it's a universe, but it can be, in the very future, if you think very, very much farther ahead, it would be interesting. Now, one thing that I want to do, and fascinating too, it would be super targeted, right? At this point, to match the person that has the same. But I think the uniqueness of the exchange between the mother and infant is going to be always something super special, right? One of the things that I want to do, and it is part of my CIHR grant, is that at the end of the experiments, I want to also collect milk from Inuit mom, because the babies from Inuit moms, especially when they're born premature, they are more prone to have NEC. Let's say I have a milk that tells me this molecule is related to NEC, and this mother doesn't have it for whatever reason. Maybe it's genetic reason. We don't know. Well, maybe we can target that baby with specific microRNA that are going to help to heal the gut. So this is how open and wide, and I would need another life, a second life, in order to get there. But let's start and continue what we have and what we understood. It's very fascinating, and I wish that very many people are going to take up on what we have found here in Ottawa and then keep going with the research. Is worth it. It's worth it.
Wow, it's fascinating. It is, as you said, it's a whole universe that just opened up, and there's so much to explore out there. Yes. Okay, my next question is, you talk about, you always talk about encouraging moms to breastfeed, offer their own breast milk. How can we continue to support parents who want to breastfeed? Because we always think that we are very supportive, but we know there's a lot of gaps to be filled and a lot of work to still do to maximize the potential for breastfeeding after the discharge from the ICU. So what is your perspective on that?
So my perspective is that what we have done, I can tell you what we have done here in Ottawa and what we are doing here in Ottawa, right, at CHEO and at the General. But the babies are born at the General, but then the babies that are transferred from other hospitals, they come directly to CHEO. And there is a lot, as you said, a lot to be done. So if the mother has a premature baby, we really inform the parents about the benefits of donor milk or the milk of the oral immunotherapy. We give them a special mini kit so they don't get discouraged even if there are only 1 or 2 drops of milk that they come out from their breast. We understand that they are in stress, that is something that they did not forecast, that they didn't expect at all. And it's also an education that we have brought into the unit. So all the mothers are offered, okay, to try at least breastfeeding. And I think this is very, very important. In the parents that arrive at CHEO with either their term and premature baby, we don't know what the other hospital has discussed. And so what we do an automatic consult on a lactation consultant. I think lactation consultants are very precious because they are really helping the whole team, not only the physician that we know, the principals, but especially the mothers that are struggling, that are putting the baby, they have sore breasts and all these things, to try to overcome this natural difficulties of the breastfeeding, and find different strategies to try to get and collect as much breast milk as we can.
Absolutely. So let's go back to your comment about the formula that is bovine based versus the human based. So where are we now in Canada with that?
That's a good question. Trying to fight. I know that there are some centers that are using exclusive milk and is used as a rescue. So there is this product that does, exclusively donor milk, and is a formula that you can add to the maternal milk. Even if you add a little bit, you can fortify it, right? With that. As far as I know, I can tell you all the things that I have seen and I read. There are colleagues of mine in British Columbia and in Alberta that they are using it, and they have reported several cases in which they were very successful, and they aim to target just a very, very tiny one, less than 750 grams. I do believe that even if it's not available right now for parents because it's an expensive product, I think we should always integrate in our decision making, talking to parents about it. And this is what we do.
Absolutely. I think education is always a key, and how can we all work together, even like educating each other as healthcare professionals on what you've been seeing, the findings, and you're sharing your experiences. And I love this platform that we have here, so we can actually come and bring this knowledge and share across Canada and many other countries what we are learning and sharing the experiences, really. But Dr. Ferretti, thank you so much. I also want to let you know that we are actually hosting 2 workshops, lactation workshops in October and November for healthcare professionals. Our goal is really to help units across the country to improve the rates of breastfeeding. So we're going to have a great lineup of speakers, and all the information is on our website. I'll be very happy to have you sharing also your perspective there. Okay. But thank you so much.
Thank you to you. Thank you very much to have me. It was a great experience.
Thank you. And to all of you watching us from home, from the hospital, on our YouTube channel or Facebook, thank you so much for joining us here today. The video of today's sessions and all the videos from our live series are available on the CPBF website, which is the CanadianPreemies.org. And I want to thank our sponsors, AstraZeneca and Water Wipes, for their ongoing support to our education sessions. And we are a charitable organization, and we believe that through education and support, we can empower families, ensuring they are ready to care for their premature babies. Visit our website, consider making a donation. Together, we can create a brighter future for all families. So I see you again next Friday, 1 p.m. Eastern Time. Take care and stay well.